Hi, my name is Irina Kunisch, and together we're going to answer the question, what is a neuron? This is a brain. The brain is responsible for controlling the nervous system. Data from the sensory organs gets transmitted to the brain, where decision-making takes place and movements are controlled. Furthermore, the brain manages the body, including all involuntary processes. An adult brain weighs around 1 to 1.5 kilograms. And while countless self-help books and apps promise to train your brain, it is not a muscle. But what then is it? In the following 10 minutes, you're going to learn about the quintessential part of the brain, about the tiny little things that make everything happen, the neurons. The nervous system is built on two basic kinds of cells, neurons and glia. According to approximations, there are about 100 billion neurons and 10 times as many glia cells in the average human nervous system. Each of the 100 billion neurons is believed to send between 5 and 50 messages, or so-called action potentials, per second. Neurons form connections that allow a transmission of impulses from one location to another. They are the functional units that have three main purposes: detecting input at their membranes, integrating this input, and communicating the results to other neurons, muscles, or glands. Like other animal cells, neurons contain a nucleus, membrane, and other typical structures. What distinguishes neurons from other cells is their unique shape. Today we are going to focus on the example of vertebrate motor neurons and their components due to their relative simplicity. A motor neuron, such as the one on the right, has its soma in the spinal cord. Through the dendrites, the neuron receives excitation and runs impulses along its axon towards a muscle. Dendrites are the tree-like branching fibers at the top of the neuron. The greater the surface of the dendrites, the more information they are able to receive. The shape of the dendrites can vary between different types of neurons and even between one and another given neuron. At the base of the dendrites we can see the soma, which contains the nucleus and other structures. The soma is connected to the presynaptic terminals via the axon. The axon is the key component in transmitting information by conveying impulses either toward other neurons or, for example, muscles. Axons can be covered by an insulation called the myelin sheath that look like threaded beads. The interruptions between the sheaths are the so-called nodes of Ranvier. An axon ends in branches which form at their ends smaller nodes called presynaptic terminals. Here, neuron-to-neuron communication is made possible. Neurons interact through membrane protein complexes. These are proteins that are bound in a structure, such as a channel. Like other cells, neurons contain ions, which are molecules that can carry electrical charges. Some neurons have transporters that can pump ions in and out of the cell, and membranes that can filter certain types of ions. These components are the basis for the electrical communication of neurons. On the left, you can see such a pump, the sodium-potassium pump. The first row shows how negatively charged sodium ions bind to the inside of the pump and then are transferred outside of the membrane. In exchange to that, positively charged potassium ions bind to the inside of the pump and get located to the inside of the cell. When a neuron is at resting state, the membrane potential is kept at 65 MV. In order to send signals to other cells, it has to get out of the resting state. If a neuron receives input in form of, for example, neurotransmitters at its dendrites, the cell can be excited or inhibited. Let's say this input is excitatory, such as glutamate. After receiving this, sodium rushes through the pump inside of the cell, which essentially depolarizes the cell. Here the membranes we already talked about come into play. Voltage-dependent sodium channels open. This elicits a so-called action potential. An action potential is basically a spike or a sharp voltage change throughout the cell. You can see such a spike on the picture of the right. An action potential can only be elicited if a certain threshold is met, which inhibits a constant firing of the neuron. After eliciting an action potential, the cell repolarizes in order to get back into the resting potential and in equilibrium. During this period, the neuron is not able to elicit another action potential. Now we are going to discover how information travels between neurons and other structures such as muscles in order to produce and control behavior. So we need to zoom into the areas where the neurons connect, the synapses. On the left, you can see two connected neurons. We call the neuron that elicits and transfers the spike the presynaptic neuron, and the receiver neuron postsynaptic. In order to transmit impulses between neurons, signals need to cross the membranes. On the right side, you can see a picture of two types of synapses: chemical synapses in red and electrical synapses in blue. Chemical synapses use neurotransmitters to bridge the small gap between two neurons. These neurotransmitters bind to receptors that then either open or close ion channels, similar to what we discussed before. Electrical synapses, on the other hand, are connected through the binding of similar channels between the presynaptic and postsynaptic cell. A small channel or pore connects the cytoplasm of each cell and allows ions like sodium, potassium, and others to flow between the neurons. Let's summarize what we learned so far. Neurons are the functional units of the nervous system. They have three purposes: transmission, processing of information, and initiation of actions via electrical or chemical communication. The most important components of neurons are the soma, the nucleus, the dendrites, axon, and the presynaptic terminals. If a neuron wants to fire, it has to get out of its resting position. Firing is also called an action potential and happens when a certain threshold is crossed. If the neuron is depolarized, sodium-potassium pumps and voltage-gated sodium channels establish an equilibrium again after a refractory period. Neurons are connected at either chemical or electrical synapses. In chemical synapses, neurotransmitters are released, and in electrical synapses, neurons are connected via similar channels and therefore allow for a direct transfer of ions. So you might wonder if this is it. Is this the big mystery of how the brain works? Just some neurons that like to fire and some channels here and some synapses there? And the answer is no. While scientists understand the brain better day by day, I want to quote Christoph Koch, who is the president of the Allen Institute for Brain Science. We don't even understand the brain of a worm. Neurons are the building blocks of our central nervous system, but we don't even know all the types of neurons that there are in the human brain. There are labs working on building something like a periodic table of the neurons, but it will take quite some time before we can fully characterize all the types of neurons there are. We do know that certain neurological or psychiatric diseases target certain types of neurons, but we don't know why and how. The Dravet syndrome, for example, a severe form of epilepsy in young children, is an example for this. If we understand why and how certain types of neurons are targeted, we could improve or even find treatments. Earlier we touched on the topic of neurotransmitters, but just very briefly. The best understood neurotransmitters are GABA and glutamate, but there are other types of neurotransmitters that have to be studied thoroughly. Antidepressants, for example, are not acting on glutamate or GABA, and yet are some of the most prescribed drugs there are today. Some people call this the golden age of neuroscience. There has never been a better time to be curious, so let's explore. I want to thank you for your attention. Really, thank you a lot. And I'll leave you with a list of my references. Bye!